So thank you very, very much. And thank you for all the speakers today. We are now moving on to the student section. So we wanted to start by a few alumni presentations. So we have 3, but we're going to have first Kelli and Samuel talk, and Jenny Chandler's is a recorded one, which we're going to show while we're dealing with the voting and the reading, because we're really, really late. So without wasting more time, I'd like to introduce Dr. Kelli Anderson. Kelli did her PhD with me, and her honors with me, and it's been a real pleasure, and we've been working together afterwards on and off, and she's now got a permanent position as a lecturer at UTAS. Kelli, please.
Hey, can everyone hear me?
Yes.
Yeah, great. I've been switching back and forth between home and office and all sorts, so sometimes my sound settings are not so great. Let me share my screen.
Perfect.
Okay. Yep, great. Okay, so I will keep it quite brief. So I'll go through and introduce myself and talk a little bit about my journey, and then I think touch on some of the things that I learnt in GeneCology and that I thought were fantastic, and then maybe some other things that maybe could be looked at a little bit more when I've gotten out and realised maybe I needed a little bit more training in some areas and things like that. Okay, so as Abigail said, I'm down at IMAS in the north of Tasmania in a lecturing and a research position here. But my time definitely as a researcher started with GeneCology. So I did my honours in some areas related to fish and oyster research in 2006, 2007. And I think that was before GeneCology really took off as we know it today. I think GeneCology started really around 2008 when I started my PhD. So I was one of the first people to use the rotogene and all that kind of thing in the lab, and we were just really getting into some of the gene expression and that kind of work in the GeneCology lab there. So pretty exciting times. My research is mainly related to questions around how animals respond to stimuli, so things like climate change, so it might be temperature, ocean acidification, stress, salinity, stress, things like that. And I'm a molecular biologist. So I also came back and did a postdoc, and that was working on Abigail's ACR project with the giant grouper, so using transcriptomic tools to study larval development and thinking about optimizing husbandry techniques and things like that. So since then, I've kind of been all over the place. I've done 4 postdocs. I also spent a bit of time working as a consultant, as a conservationist and that kind of thing. So I've worked with a few different salmon species. We can see over here, we've got coho salmon, Chinook in New Zealand and Atlantic salmon. Also worked with pearls and some other species as well, kind of all over the place really. So I've kind of already spoken about my research background, and I kind of got a bit of a shock when I was going to put a list together to tell you what I'm currently doing, and I found out that I've probably got too many projects, as a lot of academics I think find, but I'm still working on temperature, which is really the thing that I get excited about. So research to do with reproductive function still, which is my pet. Immune function in salmon, so looking at skin lesions that occur in salmon during warm summer months in New Zealand. Looking at cardiorespiratory health in farmed salmon as well, because when you selectively breed fish and you keep them in a different environment to what they're evolved to handle, you can really impact their physiology and their morphology as well. So having a bit of a look at that. Looking at transgenerational adaptation and plasticity, and all of these projects are underpinned by my favourite thing, which is transcriptomics and biotechnology. And I'm also doing a little bit of development and understanding the biology of new species that have never been farmed before, so that's kind of exciting. Teaching in aquaculture, biotech, and also on various committees and things these days, like the Animal Ethics Committee, which I actually enjoy quite a bit as well. Okay, so what am I really here to talk about? So the things that I tried to reflect a lot about this, and things that I really think GeneCology does in a fantastic way is skill development in the lab. So this is something that I really don't think I need to talk about too much with this group because I'm sure you're all well aware of the skills and things that you can develop in molecular approaches, statistics, R, and all of that kind of thing, and that's amazing, so I won't go on about that. But one thing I did realise when I kind of moved on was how well resourced GeneCology is. You know, I've been to labs that claim to be molecular labs, and then you get to some point and you think, oh, how can I actually look at my RNA quality? Oh, you can't, because we don't have any equipment to do that. So that was something that really was surprising to me, I think, when I left GeneCology as well. Something that I think, I don't know if I took it for granted, but looking back, had a profound impact on the way I developed as a scientist was the actual structure that GeneCology has, which I think is something quite special as well. So, you know, there's different students at all different levels, and you've got postdocs, and you've got people who are more experienced, and then you've got people with real niches and real expertise. So throughout my PhD and my postdoc as well, I always found that I had a lot of access to specialists and people that were more experienced that I could learn from. So whether it was learning in my postdoc, what is R, what is code, what can I use with this, what can I use this for? There was always someone around, which was really fantastic. Something I also got out of it, quite a bit out of it, was teaching and guiding other students. So I guess that's being integrated into this kind of structure where you can help other people, and that has really helped me, I guess, move into an academic career as well, because half of my job is doing that. It's teaching students, and now I've got several postgrad students of my own. So that really has set me up to move into academia quite nicely. Mentoring as well has been a huge thing for me. Yeah, I'm looking at Abigail's photo, but you can't tell, but even just having, you know, support in GeneCology as well has had a huge impact as well, and I think GeneCology really does that well. Culture also is something that I didn't realise what I had until I didn't have it anymore. Even things like what you're doing now, you have the opportunity to come together to talk to each other, to understand what each of you is researching. It seems really straightforward, but again, moving to other labs, it's not always the case. I've worked in places where you can go along for quite a while and have no idea what the people down the corridor are researching, and people don't talk to each other and they don't come together as a group. And I think creating that culture is something that GeneCology does really well. And, you know, today, of course, is evidence of that. So those are I guess, the things that I really, I guess, came up with when I was trying to reflect on my time with GeneCology. So we'll get into, so this prepared me really well to work on well-established projects, which I'll talk about again in a minute. But before we continue, I'm going to raise a few points that I think GeneCology can work on, but I also want to point out that skills are things that you learn over the course of your whole career, really. So if I bring something up, I'm not saying that, you know, you need to learn it all during your PhD or postdoc. It's an ongoing kind of process. But you can definitely plant the seeds early on in your PhD, and then you'll benefit from that later. The other thing is that things have probably changed quite a lot since I was with GeneCology. I know at UTAS, even in the 2 or so years that I've been here, things are changing rapidly all of the time to cater to the needs of the people in the group. So some of the things I mentioned, maybe you're already doing, and that's great as well. Okay, so one of the things that is every scientist's dream, I think, is to work on a really well-established project with good budgets, good administration support. And that's what I had pretty much throughout my PhD and all of my postdocs. The thing with that is that I probably wasn't very well positioned to transition to a workplace where I really had to develop projects from scratch and then manage them. And an example that might seem trivial is that I had never actually created a budget, or maybe, you know, you come up with a rough budget, but it's not really an in-depth thing that takes into account all of the complexities of running a project. And the consequence of that is that I've grossly underestimated how much things are going to cost me in the last couple of years. So I think there might be some opportunities for training there. So it might be an actual training course, or it could be something that's a little bit more informal. But something I think I might have benefited from was, even as a postdoc, is to be maybe a little bit more engaged in the inner workings of a project, to keep track of things that or money that you spend over time. But then I think the important part is to actually go back and reflect on that and see how good were your predictions versus what did you actually spend. And I think maybe that would have benefited me. So let's see. The other thing that I think is probably part of my personality as well, but I found that I probably wasn't very good at the art of persuasion. So I think as you start to transition to more senior roles, you have to really be able to negotiate. So it might be with other staff members with industry. You also have to be able to know how to resolve conflicts. And it's probably a testament to the great culture that I had at GeneCology that I never really had to resolve any conflicts. But now that I do, I think that maybe some training would have come in handy. And something that I did recently last year was the university was doing some training to do with your type of personality. And so you assess your personality, and then it gives you like you get information about how you deal with other people that have different personality types, and how to deal with other people of different personality types. And it's not a magic wand or anything, but it was enlightening and helps me to kind of deal with other people in a different way and consider approaching things in a different manner, which I think is really, really important. The other thing is grant writing, but this is probably true for everyone. I dipped my toe in during my PhD applying for travel grants, which is fantastic. But I didn't really understand that it was a real art. And I don't think I understood that there are a lot of tools out there, and simple tools that you can use to make your arguments more persuasive and more convincing. So something like the Milton Hand is something, it's like a 5-step process of persuasion that I recently learnt about. And this is an ongoing process, so I'm still learning about grant writing, but I think, you know, maybe there's an opportunity for some workshops early on. And also something simple like reading successful grants from people that, you know, have been successful within your group. Because I think I'd read maybe an FRDC or, you know, the projects that I was working on, but outside of that, I hadn't done any reading and wasn't really familiar with different structures and different ways that people put forward arguments to try to be really competitive. So something really simple like that might help as well. So I think that's probably all I was going to, oh yeah, sorry, one more thing. So the other thing is probably mental health training, which I think is important. So I'm not sure if something is available in GeneCology for that these days. But, you know, I recently did a course about responding to claims of sexual harassment. And also, as I've moved into academia, I've realised that a lot of people are struggling with some, you know, issues that maybe I'm not really well equipped to help them with, and I don't know how to help people. And that's something about having a culture of support and that kind of thing in your institute as well. So I think there needs to be some more awareness about that, which when I've moved to other institutes, maybe they haven't done so well. So this was supposed to be a 5 to 10 minute talk, so I hope I've kept to time. I didn't actually look at it. But anyway, that's a really quick rundown of some of the things that I got from working in GeneCology, which was an amazing experience, and I hope to come back one day and see you all. But yeah, if there's any questions or comments, yeah, definitely feel free to ask.
Thank you, Kelly, very, very much. You raised some important points, and indeed some of the suggestions that you made are very useful, and I took note. Some of the issues like mental health, this is something that has to be done at the university level, and
Yeah.
we raised it quite a few times, and I think they are trying, although, you know, one can always do better. I'm glad that overall it was a good experience. I think because we are running so late,
Yeah.
we might have to just keep going, but if you are staying and you're available for questions, and if people want to write questions in the chat of question answer, then we can do that. But Samuel, can we move to you? And Kelly, I'll have to ask you to stop sharing, but really appreciate your time in the middle of the day. And if you can stay with us and be with us to vote on the 3-minute thesis, that would be brilliant. If you can't, I fully understand.
No problem. Thanks, Abigail.
Samuel, would you like to start?
Yes. Can you see my screen?
Not yet. You have to press share. After
Yeah.
you see it, you have to press share at the bottom right-hand side.
Yeah, it's coming up with all my different screens. Anyway, I can just talk.
Okay. Yep, that's fine.
Yeah, it's probably easier. So thanks for having me. I was quite shocked when I was asked to give a talk. I've only just graduated in February. So I was at USC from 2017 to 2020, working with Peter Tim's group on chlamydia in koalas. I worked on a vaccine, and I was part of the GeneCology during that time. So I guess I was going to start with what I thought was good about being part of GeneCology, and then move on to where I've moved on to since then. So similar to Kelli, I found that GeneCology is really unique. I've been at a few different universities, and this is the only research centre that is really diverse and gives everyone lots of opportunity to present. I mean, I made a list of those veterinary sciences, ecology, bioinformatics, microbiology, immunology. The list continues to go on, and every single talk that you go to is different to the next, and it's really positive as a student to be able to get that involvement in all these different disciplines, and also be able to talk and practice talks for different conferences and confirmations. It's all really positive, and you always get a lot of people coming to your talks as well, which is great. You get lots of questions, which help ultimately with becoming a better scientist, I think, anyway. The other great thing I found with GeneCology was the publication award. So I was fortunate to be able to apply for the publication award twice, which then allowed me to go to 2 international conferences where I went to Germany in Dusseldorf to a chlamydia conference there, and then I was also able to go to the ASM microbe in San Francisco in 2019 as well, which is really great, which actually was able to then line up a job interview in America, and it was all because I was able to have the funds through GeneCology to be able to go. So it was amazing, and I don't know of any other research centres that offer these types of things. So it's a really positive centre to be involved in, and it's quite rare. As I said, I've been in 4 different universities over the years, and it's the only research centre that's been able to allow me to do these types of things. So as I mentioned, I was able to get a job interview, which ended up resulting in me acquiring a job at Monash Biomedical Discovery Unit. I moved into there a month after I submitted my PhD, after moving back to Melbourne from Queensland. So I thought I was really lucky to be able to have the connections through GeneCology that were allowed me to get a job interview, which also meant that I could go straight into a postdoc position. So at Monash BDI, I was working on genetic engineering of bacteriophages. This was looking at altering bacteriophages to pan-drug resistant escape pathogens. Escape pathogens are, I remember them all: E. coli, strep, Klebsiella, Acinetobacter, Enterobacter, and Pseudomonas. And these are pathogens that have been identified as pan-drug resistant and a significant threat to humans throughout the world. And there's been initiatives to introduce new alternative therapies. So we were working on changing the genetics of some gram-negative bacteriophages to Klebsiella pneumoniae, which allowed it was looking at some of the resistance mechanisms for colistin antibiotic treatment to remove the antibiotic resistance within the bacteria to allow for multi-targeted approaches, so using bacteriophages and also using the antibiotics and bacteriophages. So I was in this position for 4 months, and then I was actually offered a senior research officer position at where I was before my PhD. So this was at the Centre for Women's Infectious Diseases, working at the Royal Women's Hospital. So I ended up taking that position because it was a level B position with, they just achieved an NHMRC investigator grant. So the position is for 5 years, which is pretty amazing. I was only on a 12-month contract before that. And I moved into this position in March. I did 3 days there, and then I got sent home and I've been home ever since. So it's been a really interesting journey. But I am working on, we're part of a project that is part of a Centre of Research Excellence in cervical cancer for HPV. And the main aim of this research centre is to identify complete elimination of cervical cancer within Australia over the next 3 years. So I've come in and I've been put on as an investigator on that grant, which is really cool, I think, and we're leading up to sequencing of some specific HPV or human papillomavirus infections that are different to what we normally see in cancerous lesions. On top of that, I'm also working on some grants. I actually wrote down some of those notes that Kelli put down to get in. It's actually quite difficult to find workshops and help in writing these grants as an ECR, especially when you're working from home. Most of the workshops have all kind of grinded to a halt and you just kind of left on your own. So I'm working towards a EL1 NHMRC grant to submit in October, which is looking at immune responses to the HPV vaccine. I'm not sure if people know, but there's actually 3 different types of HPV vaccine that target different genotypes of the virus. And we still don't really know, even though it's been around for 13 years now, we still don't know completely the immune responses to this vaccine, which I find quite amazing. So that'll be part of my NHMRC grant. And I also submitted a Cancer Council grant on Monday, looking at different genetic shift within the gene regions that are targeted within the vaccine throughout Australia. So the great part about the lab that I'm in has been working on HPV research for the last 15 years. And we have thousands of samples collected before vaccinations got implemented and after vaccination. So we want to take all the positive samples that we can find and sequence those. So we've applied for this Cancer Council grant too. Well, I have applied for this Cancer Council grant. It's kind of hard moving out of a PhD and moving into being the CIA of our grants. It seems a little bit surreal still. Also on that, I thought I'd give myself a little plug. We've got a couple of PhD and master's opportunities if anyone wants to come down to Victoria, brave the coronavirus outbreaks and work on. We have 2 advertised PhDs on the Melbourne University website. One's looking at Mycoplasma genitalium and burden, understanding the burden of and consequences of infection. And then another one that I'll be a secondary supervisor on, which is understanding the cause of human papillomavirus negative cervical cancers in Australian women. So head over to the Melbourne Uni website and have a look, or if anybody knows of anyone that's looking. So it's quite hard to find students at the moment. Yeah, but yeah, thank you very much. And yeah, I didn't really have any, I thought GeneCology was great and I didn't really have any advice to new things. I thought it was all pretty good.
Thank you very, very much. It's actually really lovely to hear the perspective from people who were there and left. You know, we try really hard to make it a great environment, but it's often when people go and reflect that it makes them sort of tease out what really was helpful and what was not. And I'm glad that that's the perception. I'm really delighted that it helped you get a job, Samuel, and I'm glad, Kelli, that you have a good feeling about your time with us. So now.
Hello. For those of you who don't remember me, I am Jenny Chandler. I was part of GeneCology while I did my PhD. So I came to Australia to do my PhD at USC back in 2013, and I studied there for 4 years, and then I returned back to the UK, where I'm now doing my first postdoc, which I've been doing now for about 2 years, at the Institute of Child Health, which is part of a big hospital in London, which you might have heard of, called Great Ormond Street Hospital. I made quite a jump, not just the distance, but the science, coming from my PhD, which was looking at the genetics and the hormone functioning of what makes lobsters male or female, and whether or not we can reverse the sex to make single sex populations. So that was my PhD, which I did under the supervision of Dr. Tom Ventura and Professor Abigail Eliza. It was an absolutely incredible experience. I not only loved the research side of it, but I loved all of the other opportunities that I was offered during the PhD and being part of GeneCology. I was offered teaching experience and teaching opportunities through GeneCology, not just through my supervisors, but also through other staff members. And I think that was a really fantastic, really fantastic and important part of my PhD experience. And I really hope it's something that GeneCology is still supporting and encouraging for its PhD students. I was also supported a lot through the staff at GeneCology to help write some applications for some of the bigger things at the university. So there were FOSHI awards, there were also high degree by research awards, and my supervisors and some other members of GeneCology were really helpful in helping me sort of learn how to write and create grant applications for some of those bigger awards. So again, I hope that's something which is still happening in GeneCology, because it was really important while I was there. The other big part of GeneCology, which I used to think was really important, was the weekly meetings. So I hope they're still happening, not only just because it's great to hear about all of the research which is happening amongst the group, as well as from external speakers, but importantly because it provides sort of younger PhD students with the opportunity to present in a sort of safer space where you're presenting amongst your peers, but also academics that can give you feedback on your research and your presenting style. Which then means that when you go and present at slightly bigger conferences, it's not as intimidating as it might be otherwise. So I guess a lot of this feeds into the kind of community that I felt that GeneCology had, and I hope still has. So, and I think that's a really important part of it. We also did, while I was there, I had a couple of opportunities to do some sort of outreach work. So Abigail asked me to speak with some schoolgirls, so kind of high school, where they were choosing their final year subjects and then whether or not they wanted to come and do science at university. And I think that's really important. I think wherever we can, we should be supporting young women to make choices in science and technology, and offer them opportunities to come to university and see women doing science, see what it's like, so that they can kind of see themselves doing something similar. So I don't know if that's something which is still happening, but it was something which was really important for me and has definitely shaped some of the things which I do now at my new role. So I would say if there's any PhD students or postdocs currently in GeneCology, if that is an opportunity or something you're interested in, I would really encourage you to go out and start building those relationships and offering opportunities for young girls to come and see you and come and see the research you do. And, you could even do a sort of a showcase for them. So I think that would be a really great thing for GeneCology to keep supporting. Another thing which I think GeneCology, they may well do now, but I think is really beneficial, is a mentoring scheme. So when I was there, we had a kind of HDR community group, but there was no official mentoring scheme, and I think that's really important. So we have one in my new role, and it's really helpful. So when you come in and you're in your first year, there are lots of things along the way that every time you get to the hurdle, you think, oh my goodness, how am I going to do this? But I promise you, there is somebody a couple of years up the line who's done it and knows how to do it and knows how to mitigate it if it goes wrong. So I think that could be really great for GeneCology if you had sort of more official mentoring relationships during the PhD journey, and also through early career researchers as well. So I think in summary, my big advice from the time that I spent in GeneCology is they offer a wealth of opportunity. I was given so many opportunities, not just scientifically, but through winning, opportunities to win grants, opportunities to present, opportunities to teach, so I would suggest grasp all of those opportunities, do the 3-minute thesis, have a go, attend the HDR presentation days and have a go at doing a presentation and see if you can win a poster award and all of these things. And also talk to one another. It's such a fantastic community. There is such a, you know, diversity of research going on. Don't just go to your research-based things. Go to everybody's, listen to everybody, ask questions, talk to people, because there is a lot of support and a lot of knowledge there, and it's great fun if you get to know everybody and you're really part of the community. Like I said, I had an absolutely fantastic time, and I think it's really paved the way to where I am now and to how much I enjoy my job now. So I couldn't fault it. I had a fantastic time. So it's been lovely to speak to you. Sorry I had to send a video and I haven't been able to do it live. I just didn't want to get up at 3 a.m. in the morning. But I hope you enjoy the rest of your showcase, and yeah, thanks for having me.